In today's session, we're going to be talking about the eye. We're going to be talking about how the eye is a sense organ and how the cells of the retina in the eye act as receptors. As receptors, they detect light and act as transducers to convert that light energy into electrical energy that may be sent to the brain to be perceived as an image. Let's just check that we understand that term transducer. So a transducer is something that converts one type of energy into another. And in this case, these sensory cells are converting light energy into electrical energy. There are a few key terms that we'll be using during this session when we think about the eye. The first electrical impulse that is generated by any nerve cell in a sensory organ is known as the generator potential. The first one is known as the generator potential. Every subsequent one that is passed along the axon towards the end of the neuron is known as an action potential. So when we're talking about these electrical impulses that are transmitted to the brain, these are action potentials. Action potentials and generator potentials are really the same thing. It's just that the generator potential is the first one, and it is triggered by this transduction event, this conversion of one type of energy into another. So the eye is an absolutely incredible organ, and we could talk about the structure of the eye all day, but that's really not what we're going to focus on in this video. You can go away and you can learn the names of all the different structures of the eye, but we are going to think just about the retina, the part of the eye at the back which is detecting light energy. It's pretty incredible when you think about it. Just imagine how much of an advantage that first organism had that could see. And I don't mean see like we can see, I just mean be able to discriminate between light and dark. It's such an advantage. You can see why the eye evolved, and not just why the eye evolved, but why vision has evolved independently in so many different organisms. Approximately 96% of animal species have eyes, and the first animal eyes would have done very little except detect light, and these would have established day and night cycles and coordinate behaviour, but really complex eyes evolved quite quickly. Even a slight improvement in image quality provides a significant survival advantage, allowing for this step-by-step evolution of increasingly complex eyes. There was a really famous piece of research that was done by researchers at Lund University who wanted to find out how long it might take for a complex eye to evolve because they seem so incredible. So they started off with a flat, light-sensitive patch and gradually made over 1,800 tiny improvements. For instance, a cup shape, constricting the opening, adding a lens, till eventually they got a complex image-forming eye. The researchers concluded that these steps could have taken place in about 360,000 generations, or just a few 100,000 years. 550 million years have passed since the formation of the oldest fossil eyes that have been discovered. Enough time for complex eyes to have evolved more than 1,500 times. That is really incredible. When we look at the mammalian retina, we see a really interesting thing. The photoreceptors, the rods and the cones, are found at the very back of a series of cells. So the optic nerve actually sits in front of your photoreceptors, and then there are two more cells that the light has to work its way through before it gets to the photoreceptors. So light comes in through the front of your eye, through the cornea, travels through the pupil and then the lens and then the fluid of your eye. Then it has to go through the solid matter of the optic nerve, the amacrine cells, the bipolar cells, until it gets to the photoreceptors, the rods and the cones. This seems like a slightly odd adaptation, and surely one might think that it would be more beneficial if the whole thing was flipped over, because if the whole thing was flipped over, the rods and cones would receive more light. However, that's not the way it is in mammalian eyes, or in fact in any vertebrate eyes. But it is how it is in octopus eyes. So it's really interesting when you see a mammalian eye and an octopus eye next to each other, and it seems like the octopus eye has a much better layout because the photoreceptors are at the front, and you think, well, that's odd, isn't it? Why would octopus eyes have so much better eyes than mammalian eyes? But actually, it makes total sense when you think about it. Octopus eyes aren't required to be as great as mammalian eyes. They don't have as great visual acuity. They can't see as clearly as mammals. This means that the photoreceptors in mammalian eyes really, really need to be close to a good blood supply because they have a much higher metabolism. And the choroid layer, which is a layer at the very back of the eye, is where all the cells of the eye get their blood supply. So our layout enables our photoreceptors to be close to the choroid layer, allows them to be close to an oxygen supply and a sugar supply for a higher metabolism. Also, we have a much higher turnover of photoreceptor cells, so they're constantly being replaced. And for that, they need a richer supply of nutrients that they get from the choroid layer. So that explains why we have this interesting layout. And even though the layout that we have of the retina results in a blind spot forming, that actually isn't a handicap to us in our vision because the blind spot is in two slightly different places on both eyes, and our brain is able to calculate an image where we don't see a blank space. So even though the design of our eye looks a little bit odd, it actually isn't. So let's talk about how the rods and cone cells actually work. Rod cells enable us to see in black and white, and cone cells enable us to see our colour vision. So when we look at the structure of a rod cell, we can see that the outer segment of the rod cell consists of lots and lots of things called lamellae. These are flattened membranes. Embedded into these membranes are the photosensitive pigments, the pigments that are going to enable the transduction, so the conversion of light energy into electrical energy to occur. In rod cells, these photosensitive pigments are called rhodopsin, and in cone cells, the photosensitive pigment is called iodopsin. So let's imagine a photon of light travels through the eye and hits a rhodopsin molecule. Rhodopsin is a protein molecule. Interestingly, its proper name is a chromoprotein. Chromo is a Greek-derived suffix meaning colour. So, rhodopsin is made up of two smaller molecules called opsin and retinal. So as a protein, we have to imagine that it has this shape, this three-dimensional shape where it sits in the membrane. When a photon of light hits the rhodopsin, it causes the retinal part to change shape. It becomes a different kind of retinal. When the retinal changes shape, this causes the whole rhodopsin molecule to change its configuration within the membrane. Essentially, it starts to push against the plasma membrane. It causes a physical change in the shape of the rhodopsin, which in turn creates a pressure on the membrane of the lamellae. It's this change in shape that can trigger the formation of a generator potential and then action potentials along the axon to the optic nerve, to the brain. Iodopsin works in exactly the same way. So the breakdown of iodopsin, the change in shape, causes a force to be exerted on the membrane, which can trigger an electrical impulse to be sent along the optic nerve. Okay, so we've looked at the basics of how this photosensitive pigment responds to light. What we're going to talk about now is how we have this complex vision that we have. We can see in black and white, and we can see in colour. We can see in amazing levels of detail. But how? So, the two concepts we're going to talk about next are relative visual acuity of rods versus cones and how we see in colour vision. So what does this term visual acuity mean? Visual acuity means the ability to distinguish or discriminate between 2 points. So the greater the visual acuity, the more clear your vision is. Cones have much greater visual acuity than rods. That means that we see much more clearly in colour than we do in black and white. This is because cones have low convergence. What this means is one cone cell synapses with or connects with one bipolar cell, which synapses or connects with one ganglion cell, which sends a message to the brain. That means at that position on your retina, if a photon of light hits that position, then a message will get sent to the brain saying that they saw something at exactly that position. One cone, one bipolar cell, one ganglion cell to the brain. There's no convergence of information. They don't combine information from more than one cone cell. Whereas that's exactly what happens in rods. In rods, we have high conversion. That means we could have 2 or 3 or 4 rod cells, and the information that they receive is essentially summarized into one bipolar cell. So you might see a bit of information over here, or you might see something over here, but your brain just knows that you received it somewhere within that space. Lower visual acuity because high convergence. And what does convergence mean? Convergence means the number of cells, the number of rods or cones that join with one bipolar cell. One cone, one bipolar cell. No convergence. Rods, many rods, one bipolar cell. High convergence. So difference 1 between rods and cones is their visual acuity due to their level of convergence. And difference 2 is their sensitivity to light. So think about it. When it gets a bit dark and there's low light levels, you don't go blind. You can still see, but you can't see as well. And what you can see is more and more in black and white and less in colour. So your colour vision gets worse in low light levels. This is because iodopsin, which is the photosensitive pigment in cone cells, has a higher threshold of light at which it will break down, at which it will change shape. That means that iodopsin is only broken down by high intensity light. Any level of light below that threshold will not cause a change in conformation of the iodopsin molecule, and it will not trigger an electrical impulse to be sent to the brain. So iodopsin is less sensitive than rhodopsin. So, if one photon of light hits the iodopsin molecule, it is below the threshold and no depolarization of the cone cell occurs, so no triggering of an action potential. Therefore, that action potential, that electrical impulse, will not travel down the cone cell and will not trigger the neurotransmitter, the chemical message, to be released into the synapse, and in turn won't trigger an action potential to be formed in the next neuron, the bipolar cell, and not in the optic nerve. So no message will be sent to the brain. In order for a message to be sent to the brain, you would need enough photons of light to hit that one cone cell so that enough iodopsin is broken down, so that enough electrical impulses are travelling along the cone cell to trigger enough neurotransmitter to be released into the cleft so that it will trigger an action potential in the next cell. So it's like this build up. You've got to build up enough of an electrical impulse to release enough neurotransmitter to trigger an electrical impulse to be sent to the brain. Whereas rhodopsin has a much higher sensitivity to light. That means that you would need fewer photons of light to hit a rod cell in order to generate an electrical impulse to go to the brain to say you'd seen something, which is why we are more sensitive to black and white than we are to colour vision. Number 3, why can we see in colour? We have 3 different types of cone cells and 3 different types of iodopsin. These 3 different types of iodopsin are sensitive to different wavelengths of light and are therefore sensitive to different colours of light. So it's these different types of iodopsin and how their messages are interpreted by the brain that enable us to see these wide range of colours. So, for instance, if we just get a message from one type of iodopsin, we know it's one specific colour, red, for instance. But if your brain receives information from two or more iodopsin molecules, or two or more different cone cells, for instance, red and green, your brain is able to interpret that as a colour that is a mixture between those two. And we know that in the trichromatic theory of colour vision, so we know that in our colour vision, the colour that we see between red and green is yellow, the colour that we see between red and blue is magenta, and the colour that we see between blue and green is cyan. And if we see all three cones, if all three cones are triggered by a particular thing, we know that that is white. The next thing that we're going to focus on is the layout of the rods and the cones and how that affects our vision. So, our rods and cones are laid out very, very differently. So if we think about the very centre of our vision, this is called our fovea. In the very centre of our vision, in our fovea, we have a very, very high frequency of cones. That means that when you look directly at something, you see it very clearly and you see it in colour. So we see a great level of detail because we know that cones have high visual acuity and it enables us to see that thing in colour in great detail. However, as we move further away from the fovea, the number of cones rapidly drops. So our colour vision gets worse and worse as we get closer to our periphery, and our visual acuity gets worse and worse as we get closer to our periphery. Whereas as we move away from the fovea, the number of rod cells massively increases. So the rod cells away from our fovea have a really, really high frequency. So overall, we have many, many more rod cells in comparisons to cone cells. And we have rod cells outside the fovea and cone cells at the fovea. This affects how we see. So we see in high visual acuity, we're very sensitive to our vision in the centre, and our vision is more blurred at the periphery. But it's our periphery that is most sensitive to dim light. So when we're in lower light, we see better at the periphery of our vision than we do at the centre. Which is why if you're trying to get your key in the lock and it's a bit dark, look at it from the side instead of looking at it straight on. But what's this really odd bit of the graph between 20 and 30 degrees we have here on the graph? Why does it look like that? Why is there nothing there? There are no rods or cones. This is our blind spot. This is the area of our retina that's taken up by the axons of the optic nerve, and so you have no photosensitive cells. So this is our blind spot. The next thing I want to talk to you about is dark adaptation. Dark adaptation is the phenomenon of going from a really well-lit area, so where it's really bright, into a dark area. Initially, you just see black, you just see nothing. This is because our cones cease functioning in low light intensity. And all of the rhodopsin, because rhodopsin is so sensitive to light, rhodopsin is bleached, it is broken down in the presence of light. So when you go into that dark space from the well-lit area, all your rhodopsin has been bleached, so it's non-functioning, and you have to regenerate that rhodopsin. So the time it takes you to adapt to the dark space to be able to see again is determined by the time that it takes for you to regenerate or reform your rhodopsin molecules. Nocturnal mammals show a number of adaptations to enable them to be able to see really well in the dark. So the first adaptation that they show is that they have far, far more rods and either no or many fewer cones, because we know that rods are more sensitive to light. Cats, which are nocturnal, also have a number of other amazing adaptations to be able to see really well in the dark. For instance, they have this layer at the back of their eye that we don't have, which is called their tapetum, that actually reflects light back out, so it enables them to see that light again, which is really amazing, which is why if you see cats at night, it looks like their eyes are glowing. So let's just summarise what we've learnt about rods and cones. We know that rods are rod-shaped and cones are cone-shaped. We know that there are more rods than cones, and we know that they are distributed differently. So we have more rods at the periphery and more cones at the centre. We know that rods have a lower visual acuity due to their higher convergence with the bipolar cells. We know that rods are more sensitive to low intensity light due to the fact that rhodopsin will be broken down by fewer photons of light hitting it in comparison to iodopsin. We know that there's one type of rhodopsin compared to 3 types of iodopsin, each one sensitive to a different wavelength of light, which gives us our colour vision. So I know that was a lot of information, but that's basically everything that you need to know about the eye. Well done.